Thanks very much, Jennifer, and thank you to Jennifer, Jeff, and all the organizers for putting together this fantastic concept into a great looking conference. So I'm very excited to kick it off. I'm coming to you from Vancouver, British Columbia, Canada, which is the traditional, ancestral, and unceded territory of the Musqueam First Nation. So in British Columbia, we have had a very strange year of weather. Starting last June, we had a heat dome that produced record-breaking temperatures. 49 degrees Celsius was experienced in a small town in British Columbia, and it shattered Canadian heat records by several degrees. The next day, that small town of Lytton burned to the ground in a wildfire. That was followed by a drought, more heat, a lot of damage to trees, and a lot of other issues that we wrestled with. Then we had an atmospheric river in the early fall that caused extensive flooding. And that flooding was so extensive, it cut Vancouver transportation, ground transportation off to the rest of Canada for an extended period as the major highways connecting Vancouver were all washed out. So it's been a very strange year, and there's no doubt that global weirding, as Catherine Hayhoe calls it, is here, and trees are experiencing the brunt of this. So today I'm going to talk about conifers. And conifers are a very old group of plants, and they dominate about a third of the Earth's surface. And they are experiencing a very fast rate of change, yet they have very slow lives.
Yes, Sally, I'm sorry to interrupt you. I can see your slides, but most everyone can't. Now it looks like maybe others can. Yes. Okay, good. Now you can go again. Sorry about that.
Okay. So conifers are, most conifers that are widespread show moderate to strong local adaptation to climate. And what that means is we see quite different phenotypes coming from different climates. So for example, these are Sitka spruce trees that were all grown in a common garden experiment here in Vancouver. Their provenance of origin is listed above each representative tree. And you can see that the trees coming from the warm climate of California grew much taller than those coming from the colder climates of Alaska. And they achieve this greater growth by growing for a longer period. They are adapted to a much slower growing season. They're adapted to a much longer frost-free period. The trees in Alaska, they grow relatively little because they shut down in the summer and prepare going into quite deep cold hardiness for winter. So we have a trade-off here between growth and adaptation to low temperatures. So if we think about the possible responses of populations to climate change, this figure depicts a historic range of a species, and the dashed outline depicts a shifting climatic niche for that species. So we may have natural migration from the leading edge of the species range into areas that have become climatically favorable. We may have adaptation over generations in areas that remain within the species climatic niche, but are no longer well matched with local populations. And we expect populations at the lagging edge of the distribution to be extirpated. And so these are slow processes, especially for long-lived trees. And climate, of course, is changing very rapidly. So to give you an idea of how slow these lives are, let's express it in terms of scientist careers. So looking at E. coli as an experimental organism, Richard Lenski has been able in the long-term evolutionary experiment to complete about 75,000 generations in his career. And E. coli has about a 5 megabase genome. Many of you may work on Arabidopsis, and maybe you could complete 150 generations in a career, and the genome size there, nice, small, 135 megabases. And here is a spectacular Douglas-fir. This tree is called Big Lonely Doug, Pseudotsuga menziesii, and a tree breeder or geneticist would be lucky to complete 2 generations in a career because these trees take a while to reach reproductive maturity. And we have huge genomes. Many members of the Pinaceae have genomes around 20 gigabases. Now, the massive size of these organisms also leads to other questions and other interesting biology. So one longstanding question that's only recently been addressed is about mutation rates. So of course, plants don't have a separated germ line, and there has always been a question about whether large trees or other large plants accumulate significant genetic diversity through somatic mutations during growth. So Vincent Hanlon in our group looked at this in Sitka spruce trees. He sampled 20 trees that average 76 meters tall. That is as tall as 3 blue whales put end to end, just for scale. So these are very tall trees. There's a climber collecting, and he sequenced samples from the bottom and the tops of these trees. And when he just calculates mutation rate based on those somatic mutations, it's a very low rate per year, in the order of 10 to the minus 11 per base pair per year. And we see other plants here like Arabidopsis and rice with much higher rates. But if we look at the rate of mutation per generation, it's very high in trees. Here we have the Sitka spruce, our estimate, and here we have white oak. Now, we only detected a handful of mutations in the 20 trees that we sampled, and we only sequenced about 10 megabases of targeted sequence. But we calculated based on tree architecture that one large old growth tree could have as many as 100,000 mutations spread across its crown. Mostly of those are going to be at the end of branch tips because that's where much of that the growth accumulates. So could trees migrate quickly in response to climate change? Well, we looked at migration rates and the interaction between genetics and demographics in Sitka spruce. So this is a polar view and of North America here, and we have the range of Sitka spruce going from California up to Kodiak Island, Alaska. And Sitka spruce has only reached the furthest extent in the last 500 years. So this is still post-glacial migration. Here is a zoom in on part of Alaska. This is the Kenai Peninsula, and this is the Kodiak Archipelago, where Sitka spruce arrived about 500 years ago. So Joanne Alouette combined tree cores that allowed her to date the time of establishment of trees with genetic data. And she sampled in forests that included early colonizers on the Kodiak Archipelago, closed canopy forests that developed after those early arrivals, and the leading edge of expansion of trees dispersing out into this treeless environment. And what she found, she compared the age class structure, distribution, genetic diversity of trees, forests that have been established for a few thousand years here near Seward, Alaska, with trees on the Kodiak Archipelago. And the open bars indicate that the date of establishment and frequency of trees in an old growth forest on the Seward, around Seward, Alaska, on the mainland, and the gray bars are on Kodiak Island. And what this shows is after trees arrived about 500 years ago, it took a long time for those populations to really take off, about 200 years. And that 200-year lag corresponds to a period of low genetic diversity on Kodiak Island. So we think that it took a while for sufficient diversity to arrive via pollen, in all likelihood, for the population to really take off. So migration's a very slow process. So given these slow rates of evolution and migration, what can we do as forest managers about climate change and these species? Well, there are several things we can think about. One is we could assist the migration of species into areas that become climatically favorable. Another, which is the main thing that we try and inform with our research, we call assisted gene flow, which is selecting new seed sources to plant within the existing range to facilitate more local adaptation to climate. So here we're keeping the same species for reforestation, but we're shifting the genetics of the material. It's also important we have seed collections for ex situ conservation and monitoring of populations that may become extirpated. So assisted gene flow is the intentional translocation of individuals within a species range to facilitate adaptation to anticipated local conditions. So today in my talk, I'm going to talk about several things that we've worked on in my lab and in collaboration with other groups. First of all, what are the phenotypic and genomic patterns we see that are associated with local adaptation to climate in our conifers in Western North America? Secondly, focusing in on drought, I'll talk about whether or not we see local adaptation to drought. Thirdly, I'll look at the relationship between selection for faster growth and climate adaptation. That is, when we select for faster growing trees, do we affect climate-related traits and genes? Fourth, do phenotypes and genomic data identify similar climatic drivers of adaptation? That is, what are the climatic variables that are most important for local adaptation in trees, and can we identify those from genomic data? And finally, I will briefly talk about genetic offsets and projecting maladaptation using genomic data versus using phenotypes. Most of what I'll talk about has come out of two large-scale applied genomics projects that I have led in collaboration with others. These were called AdaptTree and CoAdaptTree. And for what I'm presenting in this talk, I particularly want to recognize Sam Yeaman, who has been a co-project leader on CoAdaptTree. But these projects have involved many universities and many people. So what patterns do we see associated with local adaptation to climate? So I'll start here with work from AdaptTree. So we obtained seed from over 250 populations or provenances per species from Western Canada for these conifers. The first is lodgepole pine, Pinus contorta, focusing on subspecies latifolia. And the second is what we call the interior spruce complex. This is a hybrid complex that includes populations of relatively pure Picea glauca, white spruce, Picea engelmannii, Engelmann spruce, and extensive multi-generational hybridized populations. So the strongest association we see over and over again is for adaptation to low temperatures. And so here on the left is a map of phenotypes for cold hardiness in lodgepole pine across Western Canada. This is the province of British Columbia. This is Alberta. And the dark blue dots indicate populations that had little cold injury in our artificial freeze tests of material from common gardens. The red dots indicate cold sensitive populations. And we see a general trend here from northeast to southwest. And that corresponds very well with low temperature climatic data. For example, this map shows the 30-year extreme minimum temperature, which is lower than -40 degrees in parts of this range. If we look at the interior spruce, cold hardiness, we see a very similar distribution of phenotypes. cold hardy in the Northeast where temperatures get very low, and less cold hardy in the Southwest. One of the interesting things that came out of this work is that we had we did exome capture on both of these species, and we had about 23,000 genes in common between them. We ran genotype environment association tests and genotype phenotype associations on low temperature related variables and on cold hardiness and timing of phenology on these materials, and identified candidate genes for climate adaptation to low temperatures. And we looked at raw correlations here and found about 260 genes in the lodgepole pine and about 450 in spruce that were associated with adaptation to low temperatures. This work was led by Sam Yeaman and Kay Hodgins, and these are raw correlations, so they will include many false positives, but we found 47 of these genes in common between these two species. And that's considerable convergence. Now, you might think, well, these are both conifers, they look pretty similar, they have needles, they're evergreen, this shouldn't be a big surprise, but in fact, spruce and pine are separated by about 140 million years of independent evolution, but they're still using this set of common genes for adaptation within species to local conditions. Sam is now leading work expanding this approach to many other species, and Mike Whitlock and Tom Booker have developed some new methods for analyzing this type of convergence. So I've been talking about cold hardiness quite a bit, but of course climates are warming. So so why would we care about cold? And the reason that we care about cold is because, well, first of all, we see strong signatures of local adaptation to cold. And secondly, we're moving trees to try to, in assisted gene flow, we're moving trees from warmer to colder environments. So we have to be aware of moving them too far, too fast, and getting cold injury. But drought is more and more of a concern. There has been a lot of drought mortality in Western North America over the past decade, and especially over the last few years. California, California's picture is pictured in here, and you can see extensive mortality, but we are seeing drought-related mortality in British Columbia as well. Here's an example, and this is top dieback of trees. We've had seedling mortality, and this is either due to the heat experienced last year or to the drought experienced in the summer, or the combination. And we're seeing interactions of drought and pathogens as well. So Rafael Ribeiro is a PhD candidate who has been working on local adaptation to drought in Douglas-fir. He has tested 87 seed lots of Douglas-fir for drought hardiness, and I'll show you some of those data. And we've also done a pooled exome sequence capture on these populations, and that includes about 10 million SNPs. And Brandon Lind is a former postdoc who has led that analysis. And Raphael put in common garden experiments for both cold and drought, but the cold results are similar to what we see for other species, but I'll focus on drought here. So here's one of Raphael's drought experiments. This included all 87 seed lots from across most of the species range, including both varieties of Douglas-fir, which I'll show you in a moment. And this is 150 days of drought to death. This is a split plot design with control, well-watered plots, and droughted blocks in the greenhouse. So Raphael used chlorophyll fluorescence as a measure of the decline in tree health towards death. And so here on this map of Douglas-fir, you can see this is interior Douglas-fir, variety glauca in purple. and coastal Douglas-fir in green, and the dots indicate the natural provenances that he tested in this experiment. The control trees maintained constant chlorophyll fluorescence across the 5 time periods of testing, but the droughted trees, you can see, started to decline in chlorophyll fluorescence. The red line here shows you the water content, the volumetric water content of those trees across the treatment. Two things you can, a couple of things you can see here. First of all, we use the slope of the decline in chlorophyll fluorescence as our phenotypic measure of drought hardiness. And you can see that the purple interior Douglas-fir, which comes from a drier, colder in winter and warmer in summer climate, had a slower rate of decline than the coastal Douglas-fir, which comes from the coastal temperate rainforest. Now, there was a substantial difference in drought hardiness between the Douglas-fir varieties, but within variety, we see essentially no population variation, no signature of local adaptation to drought in these data for the coastal variety. For the interior variety, about 13% of the phenotypic variation was due to differences among populations. But this is a much less signature of local adaptation in these phenotypes than we see for cold. And it's an interesting question why we don't see more. It may be because soil moisture availability isn't just a function of local precipitation, but it varies spatially depending on soil, and also based on topography, precipitation follows quite complex patterns. It may be because we're not phenotyping the right traits, but not, regardless of that, there is considerable within population variation for cold hardiness, for drought tolerance. So digging into that, just a couple more results there. Here we have drought hardiness, the slope of chlorophyll fluorescence on the x-axis, and here's mean precipitation on the y-axis. You can see there's a general relationship, as you would expect. Populations from higher precipitation have lower drought hardiness, but this relationship is being driven by the difference between the two varieties rather than within variety patterns. And when you look at regional differences, you find contrasting patterns. Here, for example, we see drought hardiness correlated with increased precipitation in this area, but decreased precipitation in these Northern California populations. Now, these are very small sample sizes, so you can't take too much away from this, but nonetheless, there's a difference in association there. And in some areas, we saw a stronger relationship between low temperatures and drought hardiness rather than between precipitation and drought hardiness. And here are two other regions that have contrasting associations with temperature. So depending on the scale we look at, we find different associations with climate. So Raphael has dug into, with another experiment, the within population variation. And so, conducted a GWAS on this. And so what he did here was he took 20 of those populations and he did a case control experiment where within each population, he sampled the cases, the least hardy trees, and he sampled the most hardy trees. This is like a disease approach where you take diseased individuals and healthy individuals and look for associated SNPs or genes. So he found about 1,000 SNPs associated with drought hardiness in both the coastal and the interior Douglas-fir, but only 2 of those SNPs overlapped between the 2 varieties. When he looks at transcriptional contigs or genes, he finds 8 genes overlap between the 2 varieties. So we see that these varieties, they're fairly long diverged, are really using different standing variation to deal with drought, the extreme drought that was applied in this case. But this does, the variation within populations here does suggest that breeding for increased drought tolerance may be possible. We're continuing drought work. Beth Roskally is a PhD candidate working on Western larch drought tolerance. Western larch is an interesting species, Larix occidentalis, because it's a conifer in the pine family, but it is deciduous, not evergreen, and is quite a fascinating species with a relatively small distribution. Judith Nuhu is following up with the Douglas-fir drought tolerance work, working with families from the coastal Douglas-fir and interior Douglas-fir breeding program, and also validating markers from Raphael's work. So next, I'd like to look at the relationship between growth and climate adaptation. And for this, we'll take a look back at lodgepole pine. So this is the PhD work of Ian McLaughlin. And so here, what we're looking at from a genomic standpoint is changes in the average frequency of positive effect alleles. And so we had 32,000 SNPs that genotyped well on a SNP array, and those were distributed across about 18,600 contigs. And so what Ian did is, first of all, did a GWAS and identified the 1% of candidate SNPs that had the lowest p-values that were associated with 4 seedling traits. Those traits were height growth, growth initiation and cessation, and cold injury. And for each of the 186 SNPs per trait, one allele was identified that was the positive effect allele. That is the allele that increased the trait value, increased height growth, or delayed growth initiation or cessation, or increased cold injury. So positive doesn't mean it was a good trait. It just means that it increased how the phenotypes for that trait. So this analysis is broken down between populations in southern British Columbia, which is a warmer region, and populations in central British Columbia, which is a colder region. The top 2 graphs show you height. The bottom 2 graphs show cold injury. The red bars are the average positive effect allele distribution within natural populations, and the blue bars are the distribution of those PEAs in selected populations. These were seed lots produced by seed orchards that contain selected parents after 1 or 2 generations of selection. And what you can see is that selection for greater height growth is shifting the frequency of positive effect alleles, as you would expect for height, but just shifting them a little bit across many loci. And it's also shifting the frequency of positive effect alleles for cold injury a little bit across many loci. And we don't see the phenotypic effect of this shift for cold, but there certainly is a genomic signature of that, and it may reflect the time of year that we did the cold testing. So these plots show the change in positive effect allele frequency at each of those 186 SNPs per trait. So here we have height, here we have cold injury, and this is for the warmer region in southern BC. The red, these are arranged in order of increasing p-value on each SNP, and the red SNPs are those that were actually associated with all 4 seedling traits. The blue are SNPs that were associated with height growth, and the black ones are associated with other traits, in this case, cold injury. So you can see that selection for greater height increased the frequency, as you would expect, of many of these height-associated SNPs, but it also increased the frequency of quite a few of the cold injury-associated SNPs. When we look at colder regions, the results are a little bit dampened, but we see similar results, although not as strong of results on the cold injury side. But when we look at the phenological traits, this is for when the trees start to grow, and this represents a slight delay. An increase here represents a delay in growth, and this represents a delay in growth cessation. And this was a much bigger effect in terms of the timing than the growth initiation. But you can see in the warmer region, selection for faster growing trees affects almost all of these SNPs that are associated with phenology in the warmer region. But when we look at the colder region, we see many fewer responses. These trees are tested out in these natural environments, and trees that grow fast but become very cold sensitive because they grow too long are going to be damaged by cold in those testing environments. So it looks like testing in these environments is maintaining local adaptation, but still achieving growth gains. But what this really showed us is the power of a genome-wide approach to understand the architecture of these traits and understand these correlations between growth and ability to withstand cold. Okay, so for many plant and animal species, we don't have phenotypic data on climate adaptation. And one of the really important areas of work, I think, in terms of developing management conservation approaches for changing climate is to be able to understand what climate variables are driving local adaptation, and so that we can make projections for the future for those climate variables and know what the management options are. So we've asked the question again with the same set of, or a similar set of data for lodgepole pine. We've asked if phenotypes and genomic data identify the same climate variables as being important. This work was led by Colin Mauney. It also uses the 32,000 SNPs from the SNP array distributed across those 18,000 or so contexts. And we had about 280 populations in this study and 10 to 30 trees per population. So we had a good sample size. Now, genotype environment associations are associations between allele frequencies and climate variables, and they're usually adjusted for population structure, although not always. And so these genotype environment association tests are what we use to say, do we find the same climate variables as important for, as indicated as important in local adaptation as for phenotypes. So the x-axis here is the climate variance explained by 3-year common garden phenotypes. And that's those 4 seedling traits that I talked about in the previous study. The y-axis is the climatic variance explained by principal components of 186 SNPs that are associated with environmental variables. And so each data point here is one climate variable. And this tells us whether phenotypes can predict the climate that a tree or a population comes from, and whether genotypes can predict the climate that a tree or population comes from. And so what we see here for these seedling phenotypes versus genomic data is we generally see low temperature again has the strongest pattern, but there are exceptions to that. This is mean annual precipitation being picked up in the genomic data, but not much signature in the phenotypic data. Correlations about R of 0.56, not bad, positive correlation, but certainly not perfect. But if we look at 20-year data from a field reciprocal transplant experiment, we see a much stronger relationship here, an R of 0.9. And again, the low temperature variables have the strongest signature, but we see summer moisture and summer aridity related variables showing up more strongly in the genomic data. I think this genomic data is giving us a picture of adaptation over a longer time scale. This may be picking up more extreme events that we haven't sampled in 20 years of climate in the common garden. So I'd like to move on to estimating offsets, which are predictions of maladaptation and vulnerability to future climates. And we can approach this in a number of ways. We can use phenotypic estimates. We can use genomic data to predict mismatch in the future based on patterns in the present, or we can just use climatic data to predict the extent of mismatch, but we have to make a choice of climatic variables to use in that case. And an even simpler approach would be just to use geographic variables. So with phenotypic data, here's a very simple approach to predicting the what we called adaptational lag, which is really a phenotypic offset. Here we have a cline in phenotypes. This is the date of bud set or a growth cessation in Sitka spruce versus mean annual temperature. We've got a very strong relationship here. And this is height, showing a similar relationship from common garden. And so we can look at some population and in the present occupying some climate, here mean annual temperature. And we can look at the extent of predicted warming and say, what's the difference in the phenotype between the phenotype of this population and the phenotype in a climate that has the future climate of this population? So very simple. And this is assuming that current populations are optimally locally adapted, which is also an open question. But what about when we look at genomic data? So we have some phenotypic cline along some environmental gradient, as I just showed you. It might be nonlinear, might be linear, but do we expect, if we look at allele frequencies at genes underlying those phenotypic clines, do we expect to see concordant clines along that environmental gradient, or do we expect to see localized genetic clines? So we asked this question with the lodgepole pine data. So Colin Mowney, first of all, he did a k-means clustering and identified clusters of SNPs that show similar spatial covariance patterns. So they're showing similar patterns in allele frequencies across an environmental gradient. So in this case, the environmental gradients mean annual temperature, and the SNPs targeted are those associated with cold hardiness. So they're associated with low temperatures, and the y-axis here shows the frequency of the warm allele or the positive effect allele for cold injury. And if we look at all of these, there's 6 clusters here with different patterns, and looking at them put all onto the same plot, you can see that for several of these clusters, we see little, like this black one, cluster 4 or dark gray, we see little variation in allele frequency across the cold portion of the species range, but we see a steep climb in warm allele frequency across the warmer portion of the range. The purple cline here, cluster 6, shows the opposite pattern. We have a steeper climb in allele frequencies at low temperatures, and then less variation at warm temperatures. So let's look at this. So we see different patterns in different genes. They're certainly not all concordant patterns. Let's see what that looks like on a geographic scale. These are maps. So here we have British Columbia and Alberta, Pacific Ocean, and the Rocky Mountains run up through here. And what you can see is that for cluster 4, there was little variation on the cold east side of the Rocky Mountains, but a lot of variation in warm allele frequency on the warmer side of the Rockies. Cluster 6 shows the opposite pattern. Not much variation in allele frequencies on the west side of the Rockies, but a lot of variation on the east side of the Rockies. Now, the good news, the really good news that came out of this analysis is that we find almost every warm allele in almost every population. So even cold alleles generally have warm alleles present in standing variation at very low frequency. The same is true for cold alleles in warm populations. This means that the level of standing variation is very high, but in some cases, those alleles are at very low frequency. Okay, so the final question here that I want to address is using genomic data for offsets, as has been developed in several methods. And so here we're using genomic data to develop models associating allele frequencies along environmental gradients and the turnovers in alleles along those environmental gradients. And the one common approach to do that is a machine learning software approach called gradient forest that uses a random forest approach. And this approach was first proposed by Fitzpatrick and Keller, and there's a great recent review by Capblanc and others on using these methods, but also cautions on the need to validate models that come out of these approaches. You completed your master's with my colleague Tong-Li Wang and used our adaptree data for lodgepole pine to project genetic offsets using the SNP data set that I've been talking about in the previous work. And so these maps show projections of population maladaptation under two climate change scenarios. Here we have RCP 4.5, more moderate climate, middle of the road climate change estimate. And here we have a severe climate change projection. The top graph shows a full SNP set. The bottom shows using a candidate set of SNPs that are identified as coming from genotype environment associations or GWAS analyses that are climate associated. We see, of course, stronger projections of maladaptation over much of the range, but not in northern populations, with the more severe climate change forecast compared to the less severe climate change forecast. But we also see different patterns depending on whether the full SNP set is used or whether that candidate SNP set is used in this case, but generally see that populations in the south and more coastal populations are projected to have a greater offset in the future than more northern or more interior populations. With our Douglas-fir data, Brandon Lind has been looking at offsets in using gradient forests, but he's been using phenotypic data from Rafael Ribeiro's common garden, outdoor common garden here, as a way to validate projections of that model. And the idea here is to look at the movement of provenances of lodgepole pine to Vancouver, the climatic distance that involves, and use a gradient forest model to project the offset based on that climate distance in moving the trees to Vancouver. Now, he developed gradient forest models for the coastal variety only, the interior variety only, and the full variety. This work is still in preparation, so I'm just going to show you one graph and a couple of early conclusions from that work. But first of all, if we look at the genetic offset from gradient forest, the projection using the gradient forest models developed across both varieties versus growth increment in a common garden, we see a pretty nice relationship here. And this is quite encouraging, except that that relationship is mostly coming from that difference between varieties. Now, for the coastal variety, actually, we still see a pretty good relationship there, but the interior variety, not so much. And in exploring these models, Brandon has found that the gradient forest model, if it's trained on both varieties, it really inflates the apparent fit, and it over validates, if you will, the gradient forest models of projected maladaptation. And so these analyses really need to be done within variety, and careful attention paid to the scope of populations going into the training model. Now, in most cases, gradient forest approach predicts offsets within variety better than just using climatic data. And so the genomic data does add to that, to the quality of projections. But finally, one piece of good news from this Douglas-fir analysis is that a random set of SNPs performed almost as well as a set of climate adaptation candidates, and in many cases actually performed just as well. So what this indicates is we've spent a lot of time and resources coming up with climate adaptation candidates, but a set of SNPs well distributed genome wide may give you just as good of a prediction. I'm looking forward to seeing this paper out. It shouldn't be long. It's just in internal revision right now, and I think it'll be quite interesting. So in closing, we see that local adaptation to climate in conifers is very complex, and it's highly polygenic. We see that genotype and environment association approaches are quite good at identifying the climatic drivers of local adaptation and can be used to inform practices like assisted gene flow in the absence of phenotypic data. Scale of these analyses matters, and we have to be careful about conducting analyses over too broad a scale, for example, across varieties or subspecies. And range-wide analyses may inflate apparent relationships. And the last point is, genomic data is great, phenotypic data is excellent, but expert knowledge of species remains really important for understanding relationships with climate and for developing management strategies. So knowing your organism is a key factor. I'd like to thank all of my collaborators, the people I've highlighted in this talk who have made great contributions. I didn't previously mention Mung Mung Liu and Pooja Singh, who are also postdocs on CoAdaptTree. And with that, I would like to take your questions. And this is just a shameless mention of the fact that Fred Allendorf and others, including myself, have released the 3rd edition of a conservation genetics textbook. The title has changed and the content is greatly different than the previous edition. So it's Conservation and the Genomics of Populations. And with that, I'm very happy to take your questions.